Recording now. Yes. So welcome everyone to the population dynamics meeting of today, Friday, 9th of May. And I will start introducing the speaker of today. So I will share my screen. Just a second. Okay, probably you can see full screen, hopefully. So the speaker of today is Raghuveer Parthasarathy. I hope I pronounced it correctly, from the University of Oregon, USA. The title of the talk is Gut Microbes and Their Physical Concerns. So something regarding our speaker. So I got a PhD in physics at the University of Chicago, USA, and then I went to Berkeley for postdoc in California, and now is professor at the Department of Physics at the University of Oregon. So I enjoy actually exploring living systems where physical interaction between cellular components are giving rise to dynamic structure and patterns. Among these one, actually, you can see the bacterial communities that is studies with a focus on gut microbes. That actually is the topic of today. And for doing that, we also develop and implement new optical and a large quantity of computational tools that you can find also in his website. And in the past, but probably also up to now, he had the interest in membrane physics. So with that, I would like to leave the stage to Raghu. Please, I try to stop sharing my screen, and stage is yours. Thank you for being here.
Wonderful. Thank you. Thanks for the introduction, and also thanks for the invitation. This seems, yeah, like a really nice series that you all have. Um, and yeah, I'm Raghu Parthasarathy. Let me start sharing my screen. And as Marco had mentioned, please do feel free to interrupt with questions whenever you like. Yeah, don't feel like you have to save questions till the end. And I probably won't notice like the hand raise or something like that. So just feel free to just interrupt and yell. That's perfectly fine. Yeah, so I'm going to tell you about some of the things my group has been doing, looking at gut microbes and looking in the quite literal sense of seeing what their structure and dynamics and so on are. I thought, especially given that it's a especially geographically diverse audience, I'd point out where I am. And I kind of just for fun, I took maps from the global solar insolation map. So this is like sunlight. So I'm obviously in the United States, which even more than usual is in the news for unfortunately rather a long series of embarrassing reasons, but here we are. And up on the upper left there, there's a little box that's circled. That's Oregon, as you all probably know. But then if we zoom in even more, there's the state of Oregon. The eastern side is largely high altitude desert, which is gorgeous, and the western part is basically trees. And Portland, city of Portland, which is pretty well known, is over there. And if you go about 2 hours drive south of that, you will get to Eugene, where the University of Oregon is there. So we can even further, it's a town of about 200,000 people, quite beautiful. Here's a picture I took on my bike ride to campus a couple months ago. Lots of trees, lots of mist and rain and things like that. If you're in the neighborhood, please do drop by. So what I'm going to tell you about are gut microbial communities. And I think this audience is extremely well aware that each of us carries along with us trillions and trillions of microbes, mostly bacteria, and these are a very diverse ecosystem of many different species. And they play roles in all kinds of processes, things like digestion, which is not at all surprising because most of them reside in the gastrointestinal tract, but also things like training of the immune system, the early development of organs, the progression of a lot of diseases, especially quite complex ones like diabetes and various neurological disorders, also resistance to pathogens. So because of this, again, as this audience is well aware, there's an enormous amount of investigation of the gut microbiome. But there's something quite striking about our study of the gut microbiome, which is that if we think about multi-species communities in general, whether it's wildebeests and lions and grasses on the African savanna or a tide pool ecosystem, things like that, the spatial structure of that ecosystem is really important to its function. We wouldn't be able to understand like a tidal ecosystem if we didn't know that the tides come in and out twice a day and that starfish move around and things like that. Things like spatial structure help determine how species can coexist, how they compete, what susceptibility to invasion looks like, stuff like that. And so we might want to know for gut microbial ecosystems, what is their spatial organization? How are species situated? Are they aggregated? Are they motile? Where are they? Things like that. Moreover, what factors influence that kind of organization? And this is very, very difficult to assess with standard techniques by which we study the gut microbiome because nearly everything we know about it comes from doing things like taking fecal samples and then sequencing the DNA or possibly RNA in that and learning about what species are present, what genes are being expressed, that sort of thing. So that is immensely powerful. It's through this that we've learned about all kinds of connections between gut microbes and various physiological and disease-related processes. But kind of obviously, it provides almost no temporal information and really no spatial information about what these gut microbes are doing. So we thought, can we take a kind of biophysical approach and learn something about the structure and dynamics of gut microbial systems? And as is often the case, we thought a potentially powerful approach would be to take a good model system and to directly observe it. So our model system is the zebrafish. There's an adult zebrafish shown at the upper right. uh, Upper right. We work with larvae, which so the larval zebrafish is quite transparent. It's a vertebrate, so it has a lot of physiological similarities to humans and other vertebrates, genetically tractable, and so on. And in addition, one really nice attribute of zebrafish is that one can prepare them initially germ-free, that is devoid of any microbes. And then one can add in specific species or combinations of species, controlling, composition, timing, and so on. And these so-called gnotobiotic techniques were especially developed by a local colleague here, Karen Gilliman, a professor in biology. And several of the projects, most of the projects we work on are collaborative with Karen's group, including several of the ones I'll be mentioning today. So, as I mentioned, the larval zebrafish is quite transparent, but it's still non-trivial to image gut microbes. And that's because we want to image a quite large field of view. So about a millimeter by a few hundred microns by a few hundred microns with single bacterium resolution. We want to image in 3D because we expect structure to be three-dimensional. We want to image for many hours, and so we need a method that has relatively low photodamage or phototoxicity. And we need to image fairly quickly because roughly every minute or so, the gut is doing this. Hopefully this movie is looking okay on Zoom. We should be seeing the peristaltic contractions of the gut that are transporting material down, or even in the absence of material that are occurring roughly every minute or so. So we need to be able to take a 3D image faster than once per minute. Otherwise, everything would just be horribly blurred. So the technique that makes all of this possible is light sheet fluorescence microscopy. Probably many of you are familiar with it, but if not, the basic idea of it is actually very, very simple. One does fluorescence microscopy, but instead of the standard approach with the single lens, one takes one's fluorescence illumination and shapes that into a thin sheet, and that's shown here. And then that thin sheet excites fluorescent probes in whatever one is looking at. And then one looks at the emission with a perpendicular lens. So at any instant in time, we're getting basically instantaneously a 2D slice, and then scanning in the one remaining dimension gives us 3 dimensions. So this is very powerful because it's a very fast way to do three-dimensional imaging, and everything we're exciting is being imaged. So it's very efficient in its use of photons. We have relatively low phototoxicity compared, for example, to confocal microscopy. The resolution is a bit worse, but that for the things we're concerned with, that trade-off is worth it. So we have built a couple of light sheet setups, and one of them is shown here. This is that lens that's magnifying down the sheet. This is the detection lens over here. And we have a bunch of glass capillaries. And at the end of each glass capillary is a plug of agar gel. And in each plug of agar gel is a larval zebrafish. So they're not free swimming, but they're alive. And as far as we can tell, happily sitting there in their agar gel in the light sheet chamber. So what do we see? Well, here, what I'm showing you is a scan. So the movie is a scan in the the Z dimension, going into the plane. And we imaged the entire gut, but here I'm just showing you about a quarter of it. And this is a fish that started out germ-free, so without any gut microbes. And then we added to its environment just one species of bacteria, a zebrafish native Aeromonas species, some of which are expressing a red and some a green fluorescent protein. And before we started doing this about 15 years ago or so, nobody had done any like bacteria-resolved imaging of gut microbes in any vertebrate. So we had really no idea what we would find. And even from the beginning, it was clear that it's not just a homogeneous soup, that there is actual structure. So if you notice here, there are little individual specks. Those are individual bacteria, but there are also predominantly very large three-dimensional aggregates. Some are green, some are red, but we see lots of aggregation. And that ends up actually being very common in almost everything we look at. Most of the species or strains that we've examined are aggregated, meaning that like more than 75% of the population is in dense aggregates. Interestingly, we don't in any of the like zebrafish native species we've been looking at find kind of surface attached films, which is also a bit surprising, and I might come back to that later on. So this aggregation ends up being a major factor in everything we've looked at. And I thought I'd tell one story related to that. It's a couple of years old, but it leads to some current things, and I think it's a nice story. Before going into that, does anybody have questions about the method? Okay, I will keep going then. And again, don't feel shy about interrupting. So we got interested in the question of antibiotics. And this is especially work from a student of mine, Brandon Schloman, and a postdoc in Karen Gilliman's group, Travis Wiles, who worked together on this. So it's known from studies of humans, like the human gut microbiome, from things like DNA sequencing, that antibiotics induce large and long-lasting changes to the composition of the gut microbiome. Now, that isn't surprising. I mean, they're antibiotics. Of course, they're going to affect the gut microbiome. The surprising part is that they induce large changes even at quite low concentrations, like sublethal concentrations. And such concentrations are actually often found as environmental contaminants. So this is puzzling. How could low concentrations of antibiotics be having a large effect on gut microbes? So we thought, let's take a look at that with this model system. So we decided, again, taking a kind of reductionist approach to do essentially this conceptually simplest possible thing. Look at a standard, antibiotic like ciprofloxacin. We've looked at a couple of others as well, but this is the one we studied the most. And look at what it's doing just to a single bacterial species present in the gut. And the first species I'll show you is, again, a zebrafish native species. It's a zebrafish native Vibrio species that is not highly aggregated. This is actually the most planktonic and motile species of any that we've looked at. What I'm showing here, actually, do I have a zoomed in? Yeah, good. What I'm showing here is one two-dimensional slice in real time. So hopefully this is coming across well in Zoom. You can hopefully see just like a swarm of bees, like little individual bacteria that are just zooming around in the gut. Very little aggregation, yeah, very motile species. So what happens to this one if we add ciprofloxacin at about 1/10 the so-called minimum inhibitory concentration? So first, if we don't do any imaging, just dissect out a gut, measure how much just from plating, see how many bacteria there are. If we add a 1/10 minimum inhibitory concentration dose of Cipro, the concentration of Vibrio in the water drops by about a factor of 10. But in the fish, it drops by a factor of about 100 compared to no antibiotics. So we do have a large drop in bacterial abundance. So what's going on? So imaging showed something quite striking, which is that this sort of swarm of bees becomes a bunch of like slugs. So hopefully here again, you can, what we're seeing is one slice in real time. And what you can hopefully see is that these bacteria are much less motile. And if you look carefully, they're also longer. So we have a filamentation of these bacteria and a loss of motility. So what happens then? Well, if we look over several hours, that's the lower movie here, those filamented and elongated bacteria are basically pushed by the peristaltic transport of the gut into aggregates that are pushed out of the fish through the regular peristaltic transport. We can do like live dead staining, and those expelled aggregates are overwhelmingly alive, pretty much like they are without antibiotics, or like bacteria are without antibiotics. So we have this expulsion of live bacteria. Um, so the low dose of antibiotics isn't killing the microbes, but it's altering their ability to persist in this very dynamic physical environment of the gut. Now, again, as I think several of you are very well aware, in a sense, this. If we were very clever, this wouldn't have been surprising, because this filamentation and loss of motility are common stress response to antibiotics that's been observed, for a long time in vitro. So we have this filamentation and loss of motility. But what's happening in the gut is that the consequences of these effects are, in some sense, amplified due to this coupling of aggregation and intestinal transport. So that's what the antibiotics are doing to a species that's normally highly aggregated. What happens, sorry, that's normally very motile and planktonic. What happens to a species that's normally highly aggregated? So here, what I'm showing you is just a still image of Enterobacter, sorry, a zebrafish native Enterobacter species that's our most extreme aggregator. Something like 99% of the population exists in three-dimensional clusters. So what will happen to this one? So if I were standing in front of you, I'd ask for a poll, but you guys can just, in your minds, see what you would guess. Enterobacter with weak antibiotics, would we end up seeing an increase in the population compared to no antibiotics, a small population drop, a similar response as Vibrio, so like a 100-fold population drop, or an even larger, like 1,000-fold population drop? So you can ponder that. I think there's probably some way to do a poll, but I don't know what it is. Or I've forgotten that, blocking out memories of Zoom teaching. Um, anyway, but you can have your guess in your mind. What I would have guessed is B, thinking, okay, these are already aggregated. What do they care if there's antibiotics there? But in fact, we found D, an even stronger effect than we did with Vibrio, a 1,000-fold drop in population relative to no antibiotics. So what's going on here? So again, imaging showed us something quite surprising. The upper picture is Enterobacter without Cipro, and the lower picture is Enterobacter with a 1/10 MIC ciprofloxacin. And what we find is we still see aggregates, but instead of a diversity of aggregate sizes, we see a large suppression of the small clusters. So overwhelmingly, we see large clusters now, and the overall number of clusters is much smaller as well. So what's going on here? Well, seeing this led us to realize that there's actually a much more general question that we should have thought to ask earlier, which is, okay, we've seen that many species aggregate, and we've seen that aggregation couples to transport out the gut. So how can it be that aggregating species can persist in the gut despite intestinal transport? Well, it must be that aggregating species must always be nucleating and growing new aggregates. So here's now a movie over time of Enterobacter in the zebrafish gut without antibiotics, just sort of normal Enterobacter. What we see is, large aggregates being transported out of the gut, but there's this steady, snow of new aggregates there to take its place and grow and so on. So can we turn that kind of rough cartoon into a more quantitative model of these like cluster dynamics and say, are there general rules governing these bacterial clusters? And I'll give you a sense of this picture, and some more details are in this paper that we published a little while ago. So again, kind of staring at these movies, we realized that there's basically four processes that seem to be going on over time in these gut bacterial populations. First, there's just growth. So like cell division, so we can actually watch these clusters growing over time. So in other words, going from a cluster of n to a cluster of n+1. And what's going on is basically just cell division. So something like logistic growth for these gut bacterial populations. Second, we have aggregation. So we can observe actually clusters coming together, again, driven by this peristaltic transport. Moreover, it looks like large clusters are more likely to aggregate. So we can model that as some like size-dependent aggregation kernel. One can imagine fragmentation, so clusters falling apart. What we actually find, again, just by kind of staring at things, we never see like a cluster splitting in half, for example. But we do see individual bacteria leaving clusters. So the fragmentation of individuals, which is actually probably related to cell division. So cells divide and one of the daughter cells leaves the cluster. And finally, we have that overall expulsion from the gut, just again due to transport, which again is biased towards larger clusters being transported out. So one can do simulations of these processes and also do some analytic theory related to this. You can see the paper for the details, but you can write down, yeah, kind of just kinetic equations for these different processes and ask, given all of these, what steady state population should one have? Moreover, one thing that's quite nice is that because we can image these populations, we can constrain, or in some cases, actually measure these parameters in these different kinetic equations from the experimental observations themselves. So we can measure, for example, the growth rate, the expulsion rate, and in some cases, like for like fragmentation and so on, not the fragmentation rate directly, but its ratio to like aggregation rates. So we can constrain quite a lot of this experiment. So there's a lot one can do with this. I'll give you kind of what I think is a kind of simple way of looking at all these parameters, which is to construct like a phase diagram for what the steady state population is as a function of, let's say, the ratio of growth rate to expulsion rate on the vertical axis here, and the ratio of fragmentation to aggregation. So if you have high fragmentation over aggregation or high growth versus expulsion, you'll have a high steady state population. If these are low, you'll have basically extinction. And so that extinction boundary is the magenta line here. Using the experimentally derived parameters for Enterobacter, we would put ourselves, where did my cursor go? Anyway, here in this phase diagram, which predicts a viable non-zero steady state population, as we do in fact see for Enterobacter. So that's Enterobacter, the rates without antibiotics. With antibiotics, we can again measure these rates, and what we find is that puts us kind of here in the phase diagram. We have stalled growth and especially this reduced fragmentation relative to no antibiotics, and that puts us, in the extinction regime of the phase diagram, consistent with what we see of this gigantic population loss with antibiotics. The Vibrio, the very motile species I mentioned, without antibiotics is actually off scale on this because there's almost no aggregation. With antibiotics, it puts us at a lower population, not extinction, but a reduced population there. So in addition, another thing one can do with this picture is also calculate things like what is the distribution of aggregate sizes that we should see, and we can also compare that to experiments. This I'll just give you a quick sense of, just because I do want to get to other stories as well. But what we've done is for a variety of species, measure them. We spend a lot of our time working on computational image analysis methods to identify single cells, aggregates of various sizes, and so on, which is rather challenging in these data sets because there's a lot going on. We have things like over here, the reflection of the gut from the swim bladder, which is this highly reflective orb sitting there in the fish. There's a lot of interesting challenges with image analysis there. But at the end of the day, we can get distributions of cluster sizes and measure things like the cumulative size distribution for these aggregates. And find kind of general features, like for lots of different species, a kind of very rough power law that plateaus out at larger sizes. So that tells us kind of interesting things that do map again onto these aggregation and growth models. And if you're curious about the details of that, check out our paper on this. Okay, so oh, sorry, I think I missed. Anyway, so the upshot of all of this is that we have then this. Let me go back to here. What appears to be a quantitative picture of these aggregation, fragmentation, growth, expulsion dynamics that seems to describe not only kind of normal steady state behavior of these gut bacterial communities, but also seems amenable to understanding what the perturbations of things like antibiotics do to these kinds of things. So there's a lot to do with this. And so things that are going on at the moment are looking, of course, at antibiotics and not just a single bacterial species, but multi-species communities. And what we're already seeing is, perhaps not surprisingly, differential impacts. So if you have a 5-species community, they're not all suppressed in the same way. And it's uh, hopefully at some point in the future we'll make sense of why these differential impacts occur, and are these, comprehensible in terms of spatial structure and individual response and so on. One thing we're just starting to do is look at, are there connections between these kinds of dynamics and the development of antibiotic resistance? And this, I think, becomes especially interesting given the observation that these antibiotic perturbed gut microbes are not being killed by weak antibiotics, but are being preferentially expelled from the gut. So are these providing to the environment a kind of reservoir of antibiotic exposed species that, antibiotic exposed individuals that may be, the seeds of a resistant population later that transmits to other microbes. So that's ongoing at the moment. So what we've seen with antibiotics is essentially a physical transition of motile to aggregated bacterial communities, and that kind of drives this population response in the gut. So just thinking about that transition, motile to aggregated, can we find instances of the opposite? In other words, aggregated to motile in any of these gut bacterial communities? So, as you might guess from my putting that question there, the answer has been yes. So, here I'm showing again a picture of this zebrafish native Aramona species. That was the red and green one back in that first movie I showed a while ago. If you take that same Aramona species, which is again largely aggregated, and just grow it in standard growth media like LB broth or most standard media, you do not find aggregation. So in general, there's actually a very, very poor correspondence between in vitro behavior and the in vivo behavior that we observe in the gut. So why might that be? So one can find particular media that will induce in vitro aggregation of, for example, this Aromonas species. And this was work especially from Jared Smith, who's a postdoc in our collaborators group, Karen Gilliman, who did this really interesting series of experiments. First of all, finding that you could induce Aromonas aggregation in vitro with a minimal media that has a high concentration of glicnac. Glicnac is interesting because it's a major component of intestinal mucin, suggesting that one of the triggers for the bacterial aggregation is sensing kind of an intestinal environment as represented by the mucin. So you can get this one to aggregate in vitro in the presence of this glicnac sugar. And then he did a very neat experiment, basically just passaging repeatedly the non-aggregated component of this, mostly aggregated glicnac induced aggregate sample to basically do directed evolution to find a non-aggregating Aromonas. Sorry, that might have been a little bit convoluted, but essentially a non-aggregating Aromonas. So then we, of course, thought, well, what does this look like in the gut? And then, quite nicely, it turns out that in the gut as well, this mutant Aromonas is largely planktonic, so non-aggregating. So hopefully you can see there all this little magenta speckles of this mutant Aromonas that is non-aggregating in the gut. So through this directed evolution process, we have, now a non-aggregating Aromonas species. Now that's nice, and we can, there's things we've done with this as well, showing its, aggregate distribution and all of these kinds of things. But the really surprising part is we did an experiment in which we first colonized the gut with this Enterobacter species. Again, that's our highly aggregating species that we saw before. I apologize for the color change. This is the same. In green here, we have this Enterobacter species that we've been looking at before. So we have that in the gut, and then we just add to the water this mutant Aromonas that doesn't aggregate. And now I'm going to show you what this Enterobacter species looks like. So especially if you look at the movie to the right, instead of dense aggregates, we have sparse, or we have individual disaggregated Enterobacter. So the Aromonas mutant is inducing disaggregation of the Enterobacter species. If we watch over time, if we can measure and plot the fraction of Enterobacter that's planktonic, and within a few hours it becomes over 50%, sometimes essentially 100% planktonic. So that suggests that there is some kind of chemical cue between not only within the Aromonas species to trigger its own non-aggregation, but multi-species communication between these. So you may, of course, be wondering what is the signal that's being sent to disaggregate? We don't know, unfortunately. So yes, unfortunately, I can't answer that question, but clearly it is some kind of signal being sent to disaggregate. The other thing I'll point out is that in this case, the disaggregated Enterobacter is non-motile. And so the aggregate falls apart, but it's non-motile and is also slow growing. So it also ends up being easily expelled from the gut. And the overall Enterobacter population we see, especially after several hours, is much lower than it would be without the Aromonas species there. So, so yes, we can have a disaggregation transition induced in this case by another species. What else can we see? So here's another surprise. This one is very recent, just within the last like 2 months or so. So here, once again, just to remind you, we have this Enterobacter species, highly aggregated. There it is there. If so, yeah, it's highly aggregated. We don't see a motile planktonic bacteria. Here, however, is also Enterobacter. This one is a movie just of one plane in real time. And now Enterobacter, like that Vibrio species before, is highly motile and highly planktonic. What have we done here? Well, this one actually is in a zebrafish that has been fed. Now, broadly speaking, everything that I've shown you and nearly everything that anybody has done with larval zebrafish as a model system for understanding gut microbes has been on larval fish up to about 7 days post-fertilization. And the reason for that is that up to this age, you can keep them, you don't have to feed them. They have enough yolk to persist without feeding. Beyond that, the yolk is depleted, so one has to feed the zebrafish. And that's a problem because zebrafish are very visual hunters. They want food that is moving. So they want live food. They don't grow well on dry food at these ages. And live food, of course, brings with it microbes of its own. So rotifers are a common food for zebrafish. Here's a movie of a rotifer, and there's also fluorescent bacteria in this movie. I took this one myself a couple of years ago, and I find it a lot of fun because you can see little swirls of fluid flow near the mouth of this guy as it's filter feeding on bacteria that are present. So not surprisingly, rotifers bring themselves a lot of bacteria along, which would really complicate these gnotobiotic experiments. So we spent quite a while um, developing a method to at least partially sterilize rotifers using UV irradiation. This actually started out as a pandemic project. So the first iterations of this for many, many months were in my bathroom with UV LEDs and rotifer cultures and stuff like that. The upshot of all of which is that now we can induce basically 1,000-fold reduction in the rotifer-associated bacteria while retaining motile living rotifers that zebrafish larvae will hunt. So what we are observing here is what Enterobacter looks like in zebrafish that have consumed these UV irradiated rotifers that are not bringing with them a lot of bacteria of their own. And again, yeah, so what we see is this motility transition among the Enterobacter. This, as I mentioned, is very recent. We don't know what that signal is that's inducing the Enterobacter to change. But I thought I'd put this up, especially for this audience, both because it's new and interesting, but it also really highlights that there are really quite profound morphological transitions that can occur in the gut that should undoubtedly influence things like overall population dynamics, especially things like resistance to peristaltic transport, interactions with other species, and so on. Good. Um, so let's see. I think what I will do is just show a glimpse of the last thing I wanted to talk about. So I won't show this in detail, but we've seen now uh, uh, examples of consequences of the coupling between aggregation and intestinal transport among gut microbes. So another question one could ask is, could microbes themselves alter intestinal transport? So we got started on this several years ago with actually the human pathogen Vibrio cholera, and asking the question, how does this this bacterium invade the gut? And oops, I'm going to switch forward a little. I'm going to reshare my screen in just a moment, so don't worry. Um, I guess actually I'll do it this way. Good. So Vibrio cholera, hopefully that's showing now, like many other bacteria, has a type 6 secretion system with which it punctures adjacent cells and inserts toxins. And so we asked the question of what the type 6 secretion system is doing in vivo. Now, what we observed is something rather striking. So here again, we're looking, like in that movie at the very beginning, we're looking at the gut itself. So this is just a DIC movie of the gut tissue. And this is basically what normal gut contractions look like. And in this particular fish, we are colonizing with Vibrio cholera that has a defective type 6 secretion system. But this is also what just normal contractions look like. Here is the gut in a fish with Vibrio cholera with a constitutively active type 6 secretion system. And hopefully, even by eye, you can see that the contractions in the lower movie are much stronger than in the upper movie. So we have very strong contractions. So this was quite traumatic. We showed that it was due to this type 6 secretion system, we can quantify these gut contractions. And I'm going to skip forward a little bit. We also managed to show that it depends not just on the type 6 secretion system, but on a particular part of the type 6 secretion system, the so-called actin cross-linking domain, which, as you might guess from the title, cross-links actin in eukaryotic cells. So not other bacteria, but eukaryotic cells, like those of the zebrafish. Let me skip forward a little bit. So we discovered that actually several years ago, but what that connection is between the bacterial stabbing and the intestinal contractions has been puzzling us for a long time until the very recent work of a student who just graduated, a PhD student, Julian Go, who graduated a few months ago, who did a really neat series of experiments in which she, first of all, engineered our zebrafish native Vibrio to have a type 6 secretion system lacking this actin cross-linking domain. The zebrafish native Vibrio is a nice one because it colonizes to high abundance. It lacks cholera toxin. Yeah, it's a nice system to work with. So this again is that very motile species we showed you before. So just like the Vibrio cholera, you have stronger gut contractions if you have the native Vibrio, if the wild type Vibrio versus getting rid of the actin cross-linking domain. Okay, so what is this doing? Well, for a variety of reasons, we suspected that an intermediary in this sort of communication between the bacteria and these host gut contractions are immune cells, specifically macrophages. And in part, that's because of green are immune cells in here. Anyway, in part, that's because we did some experiments in which you can deplete the number of macrophages in a zebrafish gut. And that also gives you stronger gut contractions. So the Vibrio type 6 secretion system gives you strong gut contractions. Depleting macrophages gives you strong gut contractions. And just skipping to the punchline of all this, what we realized was going on is that this Vibrio species is, through its type 6 secretion system, causing tissue damage, basically attacking the host, especially in the posterior of the gut here. And over time, sorry, this isn't playing. There we go. Over time, it's inducing a migration of macrophages to that posterior region of tissue damage, thereby depleting the number of macrophages kind of lining the gut, causing essentially the same effect as our kind of genetically induced depletion of macrophages that leads to a strong gut contractions. So I know that was a bit fast, but just to give you a bit of a picture that it looks like the bacteria themselves can induce activities or can cause activities that change intestinal motility. Okay, I know I'm going a bit long, so let me stop there and skip to conclusion. Good. So in a very broad sense, what it seems like through all of this stuff is that we think this is giving us kind of general insights into gut microbial behaviors. What do I mean by that? So all of these examples that we've been seeing, one could say, okay, maybe this is just some weird peculiarity of zebrafish, things like these bacterial aggregates, these couplings between aggregation and transport and so on. But we think these are actually quite general because these processes, things like transport of material down the gut or bacterial aggregation, these are very, very general things. I mean, intestines throughout organisms transport stuff. That's what they do. And aggregation, as this audience knows well, is a very common, thing that bacteria do, whether in biofilms or marine snow or things like that. So these sort of mechanisms of coupling between these and perturbations that alter either aggregation or transport should be ones that are quite general to humans as well and other systems. So we are looking also at a bunch of other things related to gut bacterial systems, interspecies interactions, which I briefly mentioned there, especially using the ability of uh, the amenability of the zebrafish system to provide controlled compositions of species, adding in 1, 2, 3 species like that. These notobiotic ecosystems, now that we figured out a way to allow feeding of them without radical perturbation of the microbiome, that brings in a whole other axis of looking at interactions between um, you know, food and gut microbes and those sorts of communities. We're looking at the dynamics of the immune cells themselves. Um, we're just starting to do some things looking at not just what's going on inside the zebrafish, but zebrafish behavior. How does zebrafish interact with each other? And are there connections between that and gut microbes? And finally, just as a side thing, but one I'm very fond of, I'm kind of keen on evangelizing biophysics in general, and I've written a pop science book about biophysics as well. Good. With that, I'd be happy to take questions. And yeah, I'd like to thank my group, who've been a combination of undergraduates and grad students. And there's our former lab technician in the middle over there. And yeah, I'd especially in current situations really like to thank our funding from the Moore Foundation and also the NSF and NIH who've been um, yeah, who've been great. And as you may know from a lot of current things, a lot of us are rather worried about their future. So yeah, I'm very grateful to their support. So thank you.
Thank you, Raghu, for the nice talk. So we'll